Right, hello and welcome to the fourth revision stream now. Today I am really tired, to be honest, but that does not matter because we still have some revision to do. More specifically, we've just had an exam, not an exam to say, more like, just some assessment questions on respiration and photosynthesis. Because I spent the whole week in Mallorca, it meant that there were quite a few questions which I would have got right had I learnt the content. So there was this practical which apparently we were taught about and stuff, but obviously I was off, which meant I probably missed out on 20% of the marks for that alone. There were a couple kind of obvious questions that I missed out on, things like that. So overall, I'm hoping for just under 50%, but for context, 50% is usually a B in biology. Usually 60% is well above an A. So it's not like I failed by any means. All it means is that I know what to revise. So we're going to go do some past papers. Because we actually do have an exam soon. An actual exam. So how do I do this again? Assessment resources. Oh, they've changed this website around. What the hell? I just want to find a good question paper here. A-level biology. Oh my, they've changed the website a lot here. What the hell? Okay. Mark scheme for A-level paper 1 June 2023. Yep, that looks about right. Question paper, June 2023. Okay, we found it. Let's hope this isn't locked. Nope, okay. Okay. Right, let's see how many of these we know. Give 3 structural features found in all virus particles and describe the function of one of these. Oh shit. Okay. Hold on a minute. These are new pens, so they might be a little bit rusty to begin. Virus has attachment proteins. The protein coat. The lipid envelope. The free-floating... Now, it's either RNA or DNA, depending on what type, so that's not one. There's reverse transcriptase enzymes, but that's only for HIV. Lipid envelope, protein coat, attachment proteins. Am I missing anything? No ribosomes, because a virus uses its host cell to reproduce the viral components. It attaches to the cell, it injects its DNA, and then that DNA then goes into the ribosomes where viral components are produced. I'm missing something, surely? Attachment protein coat, lipid envelope. I'm missing something, but what am I missing? Am I missing anything? Features that all virus particles have. Oh, like, you can't say antibodies, surely? Is it antibodies or antigens? Hold on, it's been a while since I've done this topic. Oh, you know what? Those are my answers. Let's just check the mark scheme. Okay, okay. Oh, okay, hold on. No, hold on. Oh, okay, hold on. This is hit hard. Genetic material, which I guess I overthought. I said DNA or RNA. That is kind of a bit of a baity question, though, no? Because yeah, it's obviously they all have genetic information, but I didn't know you could be very vague with it, though. I didn't know you were allowed to be vague. Okay. Well, that's one. Attachment protein. Yeah. Protein cotton lipid envelope. Am I misremembering what these are? Am I misremembering what the protein coat is? Is that even something we do in A level? Am I getting that mixed up? And lipid envelope, it's capsid instead. Is the lipid envelope a capsid? Hold on. The viral envelope and capsid are not the same thing. The capsid encloses the viral genetics. The envelope is a layer that surrounds it. Okay, interesting. I didn't know that. Maybe we have learned it and I forgot. No, we have done this. I remember now. Because you always draw like 2 layers to this. Okay. My bad, guys. Genuinely. I did not know that. For context, our exams were all the way back in May. So it's been at least 5-6 months before we've revised. I have not revised much since then, to be honest. So if it looks like I'm failing, it's because I am. Wow, okay. And anyways, we need to state what the purposes are. So we could say attachment proteins allow the virus to bind with host cells. I'm sure that would be allowed. Or you could say genetic information. Whoa. You're not allowed to say genetic information. You have to say genetic material. Okay, the mark scheme is taking the piss here, I feel. Attachment proteins bind to receptors. Okay, yeah. We haven't learned about receptors. Thing is, Paper 1 does drop in a lot of Paper 2 terminology. We've barely started the Paper 2 content, so it's likely that I'm going to get some of the wording wrong. The capsid protects the genetic material. Genetic material codes for viral proteins. Okay, that's easy enough then. So literally, virus, you got your attachment proteins here. You got the genetic information there, and your capsid there. Okay, cool. Back with a bang. My god. So that was 3 marks already, but I would have gotten 1 mark. 2 marks? Bruh, okay, I would have gotten no marks to that, I think. Explain why viruses are described as acellular and non-living. Acellular? Implied from the name, does that mean that... What does acellular mean in terms of viruses? Because... Does that mean... Because viruses can only... They can only reproduce via a host cell. What else does that mean? Acellular. Does that mean they have no organelles? I remember this terminology, but I don't know what it means. Okay, I know that this is right. They can only reproduce via a host cell. Acellular. Oh, come on, I know what it means. acellular. But I'm just gonna put no organelles. We'll just put that. I know it's false, but just to check. Oh, okay. No cell surface membrane. Okay. Okay, yeah, I can see how that would work. Honestly, I just didn't know what... Oh, no, I would have gotten it. No organelles. There we go. No organelles or cytoplasm, but mainly acellular means no cell membrane. Cool. Non-living, cannot independently replicate, except reproduce. Okay. So, instead of reproduce, we'll say... And we need to be a bit more specific. A virus cannot reproduce on its own. It has to replicate via host cells. Okay, I think we would have gotten 2 marks of that. Let's be fairful. Give one reason why antibiotics are not effective against viruses. Right. How specific does it want us to be here? Does it want us to talk about how they're not complementary, like the antibacterials are not complementary to the virus structure? Does it want me to say viruses are not bacteria? How specific does it want me to be here? Because I know the mark schemes can be pricks when it comes to being specific. I'll just put antibacterials only work against bacteria, not viruses. But I feel like I'm going to get that wrong just because it's going to be unspecific. Yeah, I got it wrong. Does not have bacterial structures, does not have metabolic processes, does not have a cell wall. Okay, true. So it was asking me to be really specific and technical. Okay. I guess I'll go fuck myself. Because the way antibacterials work, again, you guys have to remember, I haven't revised in a long time. Like, information's coming back to me the same time I'm getting it. Antibacterials, if we have a bacteria here, obviously with the slime capsule, the cell wall, which is made of murin. I don't know how you pronounce it. Murin? The way that it works, I'm pretty sure, is it, like, perforates the cell wall or something. It makes it, like, freely permeable. I don't recall exactly how. But yeah, no, hold on, I might be misremembering. Antibacterials stop the bacterial ribosomes as well, right? One of those two. They either perforate the cell wall, or they prevent the 70S ribosomes from, thinking. Okay, so yeah, why are they not effective? One reason why antibiotics are not effective against viruses, because viruses do not have 70S ribosomes, or they do not contain bacterial... What would you get it for saying that? Bacterial components? Yeah, do not have bacterial structures. Okay. Honestly, while I didn't get a mark for that, which is sad, I get what it was coming for. I get it. Oh, what the piss. Chitin is a polysaccharide. The chitin monomer is a beta-glucose molecule with one OH group replaced by an NHCOCH3 group. Okay. Chitin has a similar structure to cellulose. How is it similar to cellulose? So figure 1 is the monomer. Similar structure to cellulose. Is it because... I might have to improv this one. Cellulose is made of beta-glucose, which is this. That's the structure of cellulose. This structure is structure of this one is OHH, and it has N. So, how are we going to word this to get the marks? There's 3 marks here. 3 ways the structure is similar. We could say they both have a hydroxyl group. And maybe we could say the position, which I think is 1. I think you start inwards, right? Do you start inwards? So it would go 1, 2, I don't know. Oh. 1, 2, 3. I don't know. I don't recall how you do it. Okay, well, one of them's definitely that both have a hydroxyl group. You could say... You could say... Three similarities. They are both made of beta-glucose monomers. That's definitely one, but they're both made of beta-glucose monomers. You could say that they both form chains, but I don't know if that would work. The structure of cellulose is literally this. They both have an almost identical structure. I guess you could say they both form like long, unbranched polymers. I don't know what other differences. Let's check the mark scheme. That's a tricky one. Oh no, come on, really? Oh, come on, they're taking the piss. I forgot some obvious things. The 180 degrees thing doesn't make sense to me. We were never taught that, so I'm gonna just pretend that doesn't exist. Joined by glycosidic bonds. Obviously! They're both polysaccharides. Okay, you got in what's it called? In carbohydrates, it's polysaccharide bonds. In lipids, it's ester bond. In proteins, it's sorry, that's glycosidic. That's peptide bonds. In DNA, it's phosphodiester and hydrogen between the bases. Okay. So, forms straight unbranched chains. We said long unbranched polymers. Would we have gotten it for that? I don't think we would have, but we were close. It's about the phrasing. You guys have to remember I haven't done this in ages. It's about the phrasing. I phrased it badly. They are unbranched and they are long. I could have said straight unbranched chains / molecules. Okay. So if I literally said long unbranched chains, that would have been a mark. Second mark I could have gotten for saying that they both are monomers of beta-glucose. Okay. Chitin keeps the tracheae open on the tracheal system of gas exchange in an insect. Tracheae. It goes spiracles, tracheae, tracheoles. Gas exchange does not occur in the tracheae. Explain the importance of one adaptation of the gas exchange surface in the tracheal system. Oh, 1 cell thick, maybe? For a short diffusion pathway, allowing for fast movement of gas. Maybe... Because I know the tips of the... not spiracles. The tips of the tracheals are moist. They contain water for... anaerobic respiration. Of the tracheal system in an insect. 1 cell thick would be my answer. For a short diffusion pathway. I feel like I'm missing another one there that's obvious. 1 cell thick, moist on the tracheals. Or maybe that's it. An adaptation is that it branches off into tracheals, which reach each tissue of the... Let's just have a look at the mark scheme. Yes, tracheal wall 1 cell thick for rapid diffusion. Go fuck yourself. I did it. Okay. Go fuck yourself, bro. Man, the stress that these papers cause you when you know the answer, but it's worded in a way where it makes it look like you don't know what you're on about. Fuck the exam, bro. Tracheal walls, 1 cell thick, for a short diffusion pathway. Piss off, I did it. Tracheals enter... Yes. No, I wouldn't have gotten it! Because I didn't say tracheals. No! I would have... Okay. I would have gotten a mark. I would have said there, I think, just thin walls, 1 cell thick for rapid diffusion. I don't know whether in an exam I would have had the thought to say tracheals. But you always have to say that. So I would have only gotten a mark for that. Even though I did it right and knew what I was on about in my head, if I didn't properly translate that onto the page, I wouldn't have got it. So yeah, the tracheal, because let's very quickly. So it starts off with the spiracle, and then it branches off into the tracheae, and then obviously into like the tracheos. The adaptation of these, there's multiple. Obviously the thin walls are 1 cell thick. That is, there's moisture. Oh, I guess liquid would be a better word. Because, I spelled liquid wrong, no way. Liquid, because that allows during, okay, it's changes in pressure, I think. In anaerobic respiration, the water enters, which reduces it increases the volume in the tracheals, forcing more oxygen in, I believe. Lignin is a polymer found on the walls of the xylem. Lignin keeps xylem vessels open. Explain the importance of the xylem being kept open as a continuous tube. Okay, what do we talk about here? Being open as a continuous tube. Are we going to talk about the properties of water here? Because we could say that water has strong cohesion tension. And that's why, that's why, hold on, I'm trying to draw a water here. Like that, water has a strong cohesion tension, because of the weak interactions between the bonds. Would I be allowed to say that? Lignin is a polymer found in the walls of the xylem vessel. I have not done xylem and phloem in a long time. I don't recall much about them. Lignin keeps the walls open as a continuous tube. Explain the importance of it being kept as a continuous tube. I don't recall what it is about it's the xylem. The xylem, because it's important for a continuous stream of water, a continuous uptake of water, to allow for the respiration of plant cells, maybe. Let's have a look. Yeah. Cohesion from hydrogen bonds. I would have gotten a mark, maybe, or two. So I wouldn't have needed to say anything about respiration. The idea it was wanting me to talk about was the idea that it needs to remain unbroken because of this water column. This water column needs to remain unbroken because of a cohesion tension, which we talked about. And that transpiration creates tension, which pulls the water up. I might have gotten one sympathy mark there for a reference to cohesion tension. So there what I should have said is that it needs to remain an unbroken column. This unbroken column is maintained because of the weak interactions between the hydrogen and oxygen bonds. And this allows for a continuous stream of water. That creates tension when it's used. Again, a lot of this information is slowly coming back into my head. The human disease malaria is caused by infection. Let me just get rid of this first. The human disease malaria is caused by infection with a single-celled eukaryotic organism. That is a diagram of a species that can cause malaria. Other than the Golgi apparatus, name one structure in figure 2 which shows it's eukaryotic. So prokaryotes are obviously without nuclei. Why would it be a eukaryote? I'm going to say because you can see the nuclear envelope. Nucleus / nuclear membrane / pores / nuclear envelope. Yep. Describe two functions of a Golgi apparatus. Holy shit, this takes me back. The Golgi checks and modifies. So, what that basically means is it packages, I'm just going to put proteins for now, into vesicles to leave cell via exocytosis. Another one is that it combines proteins with things like carbohydrates and lipids to obviously form like lipoproteins, glycoproteins. That has to be right, yeah? Modify packaged transport proteins, form vesicles, modify lipids. Okay, so modify package and transport proteins, release vesicles. So I went way overboard here. Packages proteins into vesicles so they can leave the cell. That would have been a mark. That would have been a mark if I split them. That wouldn't have been a mark, I don't think. Oh, adds carbohydrate to, adds lipid to. Okay, so I would have had to word this in a bit simpler terms. It quite literally just modifies proteins. It modifies proteins and it packages proteins into vesicles. Final question, and then I'm going to head off. Just going to find... A student investigated the use of cinnamon oil as an antimicrobial substance. Student added 100 millimeters cubed of each bacterial culture from a glass bottle onto a separate plate. Describe the aseptic technique she should use. Okay, isn't this just health and safety shit from a practical? You would work near a Bunsen burner. Or I guess just an open flame, because that kills all airborne particles. You would flame your equipment before and after use to prevent contamination. What other aseptic technique? Eye protection's a bit of a generic one. You probably wouldn't get it for that. Maybe flame the neck of a bottle of culture. And flame the spreader before and after use. Let's check if we would have got any of that right or whether I was completely lied to. Wash hands with soap. Okay, piss off, that's obvious. Use sterile equipment. Flame neck of bottle. Work close to upward air movement. Use a sterile spreader. Place into disinfectant. Okay. So. Reject air movement, sterilize air. Convection current. That's what it's called. So would I have gotten any marks for this? Examples of sterilizing techniques such as flame or dip in alcohol and flame or dip in disinfectant and rinse. I might have not got any marks for this, even though I know what I'm talking about. You would work near an open flame to kill the airborne particles because it creates a convection current. So I might have gotten that mark. Obviously, you would flame the neck of the open bottle. Yeah. And finally, would I have gotten a mark for saying about the equipment? Sterilized equipment. I might have gotten all 3 marks. Describe the difference between genome and proteome. Okay, very quickly, the final question. I think proteome is every type of protein that can be created by is it a cell or by an organism? Genome is all of the genetic information in an organism, I think. Let's have a look. Yeah, a genome is a complete set of genes in the cell. So yeah, all the genetic information in a cell, not an organism, my bad. Proteome is the full range of proteins that a cell can produce. So I might not have gotten that, but I know what I'm on about. Genome is all of the genetic information in a cell. Proteome is all of the proteins that a cell can produce. Okay. Well then, I think that's been decent. I know we didn't get too many marks, but it's not about the marks. It's about not only remembering the information, but knowing what we're on about. It's all good regurgitating the mark scheme when it comes to exam day, but if I don't know what I'm talking about, how can I move forward? At least with a lot of these questions, even the ones I got wrong, I knew partially what to say and why I was thinking it. You know, that question about the xylem, which I did get wrong technically, I knew that it was about, you know, cohesion tension because of the water molecules, the bonds. I knew that it would be an unbroken column and that the cohesion tension would form when water would move upwards. I just didn't know how to word it. That's a big thing with biology papers, is it's about wording it well. If you can't word it well, it's, you know, you're kind of buggered. Because you could know all the information in the world, which I like to think I know quite a lot about it, but if you don't know how to word it well, or if you don't know what the mark scheme's asking, then you're buggered. Like, you saw there, like, there were plenty of questions there where I thought I knew what I was saying, but the mark scheme wouldn't accept it. They are insanely picky with what you can and can't say. And it kind of sucks, but that's just how it is, unfortunately. You know, some questions you'd get a mark for saying something obvious. Other times, you're not allowed to say something obvious. You know, it's like, oh, state the differences between these. You state the obvious differences, you get marks. Other times you state the obvious differences. And no, you have to think outside the box. It's so annoying. And yeah, that's why I'm probably going to start to do more of these streams. Because once we clear through every past paper question from A-level, we could maybe go look at some specific kinds of questions and things like that. So yeah, hopefully that'll be tomorrow.